a pleasure for me to give this tutorial in this Dynamic Days conference. And I'd like to recall that it's not an original talk, it's a tutorial, not only for students, but for a larger audience. But still, I will begin and sometimes return to very well-known things to make it clear for the audience. So the topic of this tutorial, reaction diffusion equations in biomedical applications. And in fact, there will be two parts. The first part a little bit less, the second part a little bit larger. The first part will be a short introduction to the theory of reaction diffusion equations. Of course, it's a huge theory, and it is impossible to discuss all that in detail. It will be some questions of the theory, introduction to them. And in the beginning, I will recall some applications where reaction diffusion equations are used. And then two more specific questions in this more mathematical part will be about existence and stability of some particular types of solutions, about reaction diffusion waves, and about pulses. And then in the second part of this presentation, we'll use these mathematical results and these solutions to study some applications. And here you see a short list of them. Of course, there are many others, but time is limited. So what I choose here, it's about atherosclerosis, blood coagulation, viral infection, and immune response. And maybe also we will have some time at the end about neural field models. And we'll see how these reaction diffusion equations and these particular types of solutions are used to study these problems. So first of all, very shortly about reaction diffusion equations. These are equations of this form written here. In fact, mathematically speaking, it is a semi-linear parabolic equation. This diffusion term here can describe heat or mass production. Heat or mass diffusion, or in population dynamics, it describes random motion of individuals, for example. The function u itself, it can describe, again, depending on applications, it can be some populations, density of some populations, it can be some concentrations, and so on. And finally, this nonlinear term, f of u, usually describes heat or mass production in chemistry or chemical physics. It can be reproduction rate in biology or population dynamics. So this is the object we'll discuss. And of course, it can be just one equation as written here, or it can be systems of equations. And there are other possible generalizations. We will shortly discuss it later. So the theory of reaction diffusion equations, it becomes already rather old. It will be soon about maybe 100 years. Basically, in the 1930s, there were some works before, but basically, this theory appeared due to the works by Semyonov and Frank Kamenetsky in heat explosion. Then, at the end of the 30s, there were famous works by Fisher, KPP, it's for Kolmogorov, Petrovsky, and Piskunov, for the propagation of dominant gene, and also the works by Zildovich and Frank Kamenetsky in combustion theory. And the third large direction a little bit later related to pattern formation, and of course, everybody knows this Turing work in 1952. And then very briefly, just to recall you about these three main directions, and they continue. In fact, they appeared in the 30s or in the 50s of the last century, but they determined really the development of the whole this big theory, and in particular, the theory of heat explosion. Just only one slide here. I will not talk about it today, but the model suggested by Frank Kamenetsky is this parabolic equation for the dimensionless temperature. And from the mathematical point of view, we study here existence of solutions, stability of solutions, and bifurcations of solutions. But I think it's a very good example to show how mathematical equations are related to reality. In particular here, if this equation doesn't have a stationary solution, then the temperature will have unbounded growth and it will lead to heat explosion. So we have here a very clear direct relation between mathematical results and physical applications. And of course, this the theory was developed almost 100 years ago, but of course, we know many examples. It remains very important now. Well, another important application and the origin of the theory was related to this KPP-Fisher equation, how it is called now. Again, this is this semilinear parabolic equation, reaction diffusion equation. This time, we look for solutions like z, u of x and t equal wx minus ct. Solutions like that called reaction diffusion waves or traveling wave solutions of parabolic equations. And these solutions are shown here. It's a solution which propagates with a constant speed c and with a constant profile like shown here. And we will return to this question later. Of course, it depends on this function f of u. In this form, it was considered in these first works, but then many different functions were also considered later. And among applications, very well known, often cited applications of reaction diffusion waves, in particular, it is related to ecological invasions. One famous example is shown here. In the beginning of the 20th century, 1905, this American muskrat was brought to Europe, somewhere not far from Vienna, and it was brought here. And you see from year to year, the area occupied by this species became larger and larger. And in fact, of course, because of the spatial heterogeneity and not exactly circles, but if we do modeling, it will be like circles propagating like in space, concentric circles. It's how the area occupied by this animal grows in time. Another, of course, well-known and very actual application is the epidemic spreading. Here, it's also a classical example how influenza epidemic propagates in the United States. You see at different moments of time, like a wave from the left to the right here. Of course, there are many other examples, not only in biology, in chemical physics, like combustion agents, and also it's combustion waves propagating, also described by reaction diffusion waves, or forest fires, like in the right image here, also described by these models and by these equations. Of course, very important application in biomedicine is related to tumor growth. In fact, tumor growth, it's also an example of reaction diffusion waves. And here, of course, this reaction diffusion equation shows here in the slide, it's a very simplified model. But even this very simplified model already can be used to describe the growth of the population of malignant cells. And again, in this bottom right figure, you see these are experimental data, how tumor grows in time. And again, up to some heterogeneity, these are more or less concentric circles corresponding to the area occupied by the tumor. Another very interesting example and applications are related to excitable media. I will not talk about it today, but there are many very, well, at least, in fact, maybe I will a little bit in relation with neural field models. But here you see several examples like Belousov-Zhabotinsky reaction, famous reactions discovered, well, first by Belousov in the 50s, then repeated by Zhabotinsky in the 70s. It's oscillating reactions. And then in biological applications, it's waves in the heart tissue or waves in the brain tissue, also very well known and very interesting questions. And finally, to end with a short introduction to reaction diffusion waves, of course, another very interesting, very well-known question related to pattern formation and Turing structures. For example, here there are several examples of patterns for the animal skin. There are many other examples of pattern formation described by Turing structures. But again, I will not discuss this question today. So now, after this short introduction, I will also shortly discuss some a little bit more mathematical questions about existence and stability of some special type of solutions, in particular about reaction diffusion waves and about pulses. So here is our equation, reaction diffusion equation. We consider it in this form. Traveling wave solution, as we just discussed before, u of x and the t, it's w x minus ct. This function depends on this combination of variables, x minus ct. C is the wave speed. The wave speed is unknown. So again, mathematically speaking, we consider this problem written here, and we should find the values of C, the values of this constant in such a way that this problem has a solution. This is a mathematical formulation of the problem of existence of reaction diffusion waves. And the answer, the solution of this problem depends on the form of this nonlinearity. Some typical examples shown here in the bottom, what they often called like monostable or bistable case or nonlinearity. Why this terminology? Because if you consider this equation written at the bottom right, du dt equal f of u, then we just consider the stability of this stationary point u equals 0 or u equal 1. And it can be easily see, for example, that here u equals 0 is unstable, u equal 1 is stable. So this is why it is monostable case. And in the second case, u equals 0 is stable, u equal 1 is also stable. In this case, we have this bistable case. And I will shortly recall, of course, all these results are very well known and quite simple. I will briefly recall what happens in this case. And we will begin with this monostable case. The function f of w is shown here in the right top figure. It is a positive function in this interval, having 0 at the end of these intervals. And we look for solutions like this, like here, w of x, like a decreasing function of x with the limits 1 and 0 at infinity. So how we can study the existence of solutions like that? In fact, for one single equation, it's a very simple question. We can reduce this second order ordinary differential equation to these two first order equations. We have this very simple dynamical system on the plane. And these limits at infinity give us two stationary points, 1, 0 point here, 0, 0 point here, and we look for a trajectory connecting these two stationary points. This is our mathematical problem, and it can be easily solved. We should consider this linearized system of equation, study the stationary points. Of course, I will not do it. It's just a very simple and standard exercise. And like that, we can determine that this point, which is 1, 0, it is a saddle point. The point 0, 0, it is a stable node. So what happens here, if we study the direction of the vector field, you see on this bottom right figure, here, these trajectories go down. At this line, they go up. So the trajectory which leaves this saddle point, necessarily it will come to this other stationary point. So this is a very simple proof of wave existence. This proof was suggested already in this KPP work. And of course, it is so simple if we consider only one equation. But if we consider a system of equations, this method becomes either not applicable at all, or it becomes much more difficult, and there are some other methods which should be developed. But here, in this particular case, we obtain a very, very clear and very nice result that solutions like that, in fact, reaction diffusion waves, exist for all values of the speed greater than or equal to some minimal speed C0. So please let us keep it in mind that in the monostable case, these solutions like that, reaction diffusion waves, exist for all values of the speed greater than or equal to the minimal speed C0. And the value of this minimal speed, sometimes it can be analytically found, sometimes not. We'll discuss this question a little bit later. So we started the wave existence in the monostable case. So what happens in the bistable case? So the beginning is exactly the same. So we look for the trajectory connecting these two stationary points. But the difference now is that these two stationary points, both of them are saddles. And this changes a little bit the existence results and the proof of this existence result. But again, it remains very simple. We have this one trajectory leaving this stationary point 1, 0, another trajectory coming to the stationary point 0, 0. But in general, these are two different trajectories. We need to have the same trajectory. So what we do, we change the value of speed. C, we consider now this as a parameter, unknown parameter. We change the value of this parameter, and we can easily verify that this trajectory change, like shown here, this trajectory goes down, this trajectory goes up, and for some unique value of C, they intersect and they coincide. So like that, we obtain, again, very simple and very nice result that solutions like that, reaction diffusion waves, in the bistable case, exist for a single value of speed C. So again, let us keep in mind this result. Its classical result is the theory, and it's very useful in applications. Of course, it should be noted that here we consider just this function f of w of this particular type, where it is negative here, positive here. So this result on wave existence depends on the form of the function f, and I will return to this question later. And here are some examples. In population dynamics, rather often we have functions like that, shown here, top right, or functions like that in the middle. In the first case, it is a monostable case. In the second case, it is bistable case. And as we discussed, in the monostable case, waves exist for all speeds greater or equal to the minimal speed. And some applications I mentioned before, like cancer, tumor growth, or ecological invasions, they are described by this monostable case. But in some other case, it can be this bistable case. In this case, only a single wave exists. Well, I present here another example from combustion theory, but it is less important for us today because we will discuss only biological, biomedical applications. Now, another type of solutions called pulse solutions, pulses. So what it is? So we have the same equation, and we assume that f of u is like that. It is a bistable nonlinearity, bistable. And we look for solutions shown here at the left figure. So it is a positive solution with zero limits at infinity. So the question is whether such solution exists. And if we use the same method as before, we have the second order equation, but a little bit different. Before, for the waves, we had here this first order term plus CW prime. Here we consider just a stationary solution of this equation. We have this equation, and in fact, we can even analytically solve now this problem. And we look for the trajectory now like that. Now it is not a heteroclinic trajectory. It is a homoclinic trajectory shown like this. And we can easily prove the existence of solutions like that in the bistable case. And the result is, in fact, that such solution exists if and only if the integral of this nonlinearity is positive. You should also keep in mind that the condition that the nonlinearity is bistable is important. For the monostable nonlinearity, solutions like that do not exist. Well, and again, well, there are some examples. In the monostable case, the solution does not exist. In the bistable case, such solution exists. Well, we formulated this condition on the integral. And here, I give also another example, very interesting example of non-local equations. It's a little bit different. Now, instead of this 1 minus u, we have here y minus integral. Such equations also appear in applications. We'll see it later today. And what is interesting here, that in this case, if we consider such equation, then again, we can prove that solutions like that, pulses, they exist. But in fact, such solution is not unique. In this particular case, it is possible to have two solutions, either zero solutions or two solutions. And if we have this case of two solutions, one of them becomes stable. We'll discuss it a little bit later, just in the next small chapter of this lecture, that solutions like that, these pulses, they are not stable for the usual equation, but some of them become stable for this non-local equation. Just to finish this small part about the existence of solutions, of course, there are many generalizations and many developments related to systems of equations, to multidimensional equations, to non-local equations I mentioned, delay equations, so on. So there are many interesting questions. Some of them are open, and many of them are related to various applications. Okay, so the next just section of this theory of this lecture is related to stability of solutions. And first of all, we need to introduce here the notion of stability with shift. And to introduce this notion of stability with shift, we should remark that in fact, solution W of x of this equation is invariant with respect to translation in space. This means that not only Wx, but Wx plus h for any h is also a solution of this equation or of this problem. So in fact, we have not a single solution, but we have a family of solutions. And here this notion of stability with shift appears. And stability with shift means that if we have our initial value problem, and we have Wx is our wave solution, we have a small perturbation here, then the solution may converge not to the same wave, not to the wave Wx minus Ct, but to the wave x minus Ct plus some h. This means to some shifted wave. This is what is called stability with shift. And why such stability appears here, stability with shift? Because in fact, this linearized operator, if you look at this operator L, it's a linearization of this equation, so it can be easily verified that this operator has a zero eigenvalue, because just if we differentiate this equation with respect to x, we can see that W prime is a solution of this equation L W prime equals 0. Then this linearized operator has a zero eigenvalue, and this is why, in fact, instead of the usual stability, we have the stability with shift. Okay, and now I will present a very simple proof of wave stability. And this very simple proof is based on what is called Crane-Rootman theorem. It is a famous theorem, and in fact, it is a generalization of Perron-Frobenius theorem for matrices. So if we take matrices with non-negative, or let us take for simplicity positive off diagonal elements, then we know from the Perron-Frobenius theorem that the eigenvalue with the maximal real part for this matrix is real, and the corresponding eigen vector is positive. And in fact, the same result remains valid for such eigenvalue problems for differential equations under some conditions, of course. And in fact, if we consider such second order operator, we consider the eigenvalue problem, then the principal eigenvalue is the eigenvalue with the maximal real part. It is real, simple, and the corresponding eigenfunction is positive. Of course, it is a very useful result, but it is valid, of course, only under some conditions, in particular for the single equation, but in general, not for systems of equations. So now we can apply this result to prove the stability of waves. So here is our equation, W second derivative CW prime plus FW equals 0. WX is the solution of this equation. So as I mentioned before, if we differentiate this equation with respect to X, we have this, what is written at the right. So we have this U second derivative CU prime plus F prime U. And this equation has a solution W prime. It's a direct and very easy to verify. This means that the operator LU written here, it has 0 eigenvalue, and the corresponding eigenfunction W prime. So again, 0 is an eigenvalue, the corresponding eigenfunction U of X equal W prime. And if our Wx is like that, if we consider monotone waves, it's a monotone function, so W prime is positive. So now we apply this Crane-Rootman theorem, which tells us that the only positive eigenfunction corresponds to the principal eigenvalue. We conclude that 0 is the principal eigenvalue. All other eigenvalues lie in the left half plane, And well, in fact, the whole spectrum. I don't discuss, of course, all details here. We should remember that we consider elliptic problems in unbounded domains. We have here essential spectrum. We should also verify the location of this essential spectrum. We don't discuss, of course, this now, but here we obtain just a very simple result, a very simple consequences of Crane-Rootman theorem, which tells us that 0 is the eigenvalue with the maximal real part. All other eigenvalues have negative real parts. Therefore, we have here this stability with shift. So the conclusion is that monotone waves are stable. Well, there are, of course, the stability result is interesting by itself. There are different applications of the stability result. In particular, we can, from this local stability, we can obtain global asymptotic stability. We can use this result on global asymptotic stability to determine the wave speed. But I will not stop on that now. And another interesting application of the same theorem is that if waves are stable, then pulses are unstable. This is also a very simple application and very useful. So let's see what we have here. As before, we have this equation. For generality, I write it here with a C because it also applies for waves. We differentiate. We have this equation here. As before, U of X equal W prime is the eigenfunction corresponding to the 0 eigenvalue. And now, since we consider these pulse solutions, this pulse is not a monotone function, so W prime changes sign. So the eigenfunction U of X is not positive. So we conclude that 0 eigenvalue is not the eigenvalue with the maximal real part, because the eigenvalue with the maximal real part has positive eigenfunction. since here eigenfunction is not positive, then 0 is not eigenvalue with the maximal real part. Then there is an eigenvalue, then there is a positive eigenvalue. If there is a positive eigenvalue, then the solution is not stable. So as before, we have a very simple consequence of this Crane-Rootman theorem, which tells us that pulses are unstable because they are not monotone, and reaction diffusion waves, in fact, there are also waves which are not monotone function of x. In this case, they are also unstable. And to finish this, well, first theoretical part of this lecture, just to recall that just what we discussed before, it was for the single equation. All these results remain valid also for so-called monotone systems of equations. What means monotone systems of equations? If we, now it is U is a vector here, F is also a vector. So if we take this derivative of diagonal elements of this Jacobian, we suppose that they are non-negative. If they are non-negative, then it appears that we can apply the maximum principle and comparison theorem to this class of systems of equations, because in general, they are not applicable for systems of equations. If this condition is satisfied, then they are applicable. And then, similar to the single equation, we can study all these equations. In the bistable case, we can prove the wave existence, uniqueness. We can prove local and global stability. We can obtain the minimax representation for the wave speed. And in the monostable case, similar to the single equation, the waves exist for all values of the speed greater or equal to this minimal speed. So I have finished with this first part of my lecture, mostly theoretical part, and then I will switch to another slide, another file, if I can. As usual, something doesn't work here. Let me see. Okay. Okay, so I will share the screen again.
Maybe if now, because if you agree, Vitaly, or if some people have questions right now, maybe if you change the part.
Yeah, please. If you have questions, we can make a short break just to breathe, and if you have questions, we can shortly discuss them, and then I will continue with the applications of these mathematical results.
So, is there any question on the first part of the talk?
Hello.
Yes. Who is speaking?
This is Moitri Sen.
All right.
Yes.
India.
Hello, Moitri. Yes.
Hello. I just have one question. That you talked about stability of waves and pulses. Can you please just tell me the mathematical? What are the mathematical importance? What do we mean by the stability and instability of pulses and the waves? Is it the same as we talk about the equilibrium points, or it has some other meaning?
Well, it's okay. So I will shortly answer. Well, in general, the notion of stability here is, well, not exactly the same, but close to stability of stationary points. But the idea behind is, if we take a stationary solution, it can be, for example, stationary point, or it can be a pulse solution, or it can be a wave solution, and we give a small perturbation, and then we consider what happens with solution of this problem. So, and there are different possibilities. Either the solution returns back to the stationary solution. In this case, we say that the stationary solution is stable. Or another possibility, the perturbation will grow and the solution will not converge to the stationary solution. In this case, that this stationary solution is unstable. Is the answer is clear?
Okay, yeah, a little bit. I'll check it on again.
Okay, so of course, it's a very short answer. There are many details and many different cases, but we cannot discuss all that now.
Yeah, sure.
Okay, if no other questions, then I will. Yes, then
maybe I can ask
sorry.
one. Can I ask a
please.
quick one? Yeah.
Yeah,
Okay.
please. Yes.
Am I right that in applications, the systems that you consider are likely to be subject to external drives or that are possibly time dependent and so on? So do the solutions still look fairly simple, or do they become very much more complicated when subject?
Yeah, of course. Well, it's a general question, so my answer will not be very precise. Yes, we can consider such equations, reaction diffusion equations, with external forces, or in other words, with time or space dependent coefficients, or time or space dependent nonlinearity. There are quite many works on that, and if we talk about, for example, a reaction diffusion waves, and if we have this, for example, space-dependent perturbations, then we will not have this wave with a constant speed and a constant profile, but we'll have what is called sometimes generalized traveling waves. It's something which propagates, and the profile can change a little bit, the speed can change a little bit, but in principle, it is kind of propagating solutions.
But these are still
course,
the dominant solutions that one sees in applications.
Yes, but of course it depends on particular problem. But in principle, yes. In principle,
thank you.
All right. So, yeah. Okay.
Okay. So I continue.
Yeah, I think we can continue a little bit and.
Yeah. Well, I will stop. So we decide to make a break after 50 minutes, right? Or like that.
Yeah,
so I will...
I think that's good if you're okay with that.
So yeah, it's perfect for me.
Okay.
We will do that. I will just stop after 50 or 55 minutes. We'll see.
No, sorry. No, we will have to do a small break. If I think
yeah.
Okay, 15. Okay. All right.
Okay.
That's good.
Okay. So and now we'll see how these solutions and some other solutions and this, all that.
Vitaly,
yes.
Sorry, can you put full screen?
Yeah, I will.
Okay.
I do. Yes.
Okay.
Yes, here it is.
Yeah. Thank you. Much better.
Okay. So, in fact, again, there will be two parts in this. Applications, and the first from these two parts is related to cardiovascular system and diseases, and in particular here we will consider in more detail two questions related. The first one to atherosclerosis, and the second one to blood coagulation and thrombosis. And what I will try to do now is to put it in a little bit more general context. Considering each particular biological or biomedical question, I will start with a physiological background to explain, in fact, what is the process, what we want to describe. Then we will consider some models, and we will apply some of these results we just discussed. But then what I want also to show, just not to reduce this discussion to these particular models and these particular solutions, but to show it a little bit larger, to briefly show some other methods of modeling, some simulations, to give you just a more general idea what happens in this field. I think it might be more useful and more interesting. So let us start with atherosclerosis. What is atherosclerosis? It is a chronic inflammation which happens in blood vessel walls. Like, for example, here shown in the middle image, it's a coronary artery in the heart, and the inflammation starts here. the vessel walls becomes thicker and thicker, as shown in the right image. You see it's a cross section of this vessel. Normally, vessel walls are quite thin, like a dark gray, this area. But because of this atherosclerosis, the walls becomes much thicker, and the lumen is the part of the vessel where the blood flows becomes narrow, or in some extreme cases it can be completely occluded. And we understand that this is very dangerous, in particular if it happens in the coronary artery. It can lead to heart attack, it can lead to heart arrhythmia, and to many other situations, to clot formations. And this clot formed in the heart can go to the brain, can provoke stroke, and many other things. So now I will show you a very short movie from YouTube to explain what happens here. You see, it's blood flow here, erythrocytes, monocytes. So cholesterol penetrates vessel wall, provokes inflammation, then immune cells come there. Okay, so what is explained there? What happens? You see here in this top left figure, it is blood vessel wall, which is shown here, and low density, it is called LDL, low density lipoproteins, or rather often we call it bad cholesterol. It penetrates here, the vessel wall, it initiates inflammation here. Then because of this inflammation, blood cells called monocytes, they penetrate here. They become first macrophages, then become foam cells, and it appears that they increase this inflammation even more. So it becomes a self-amplifying positive feedback loop between of this inflammation, and more cells come in here, and this lipid accumulation, and this is why, because these vessel walls become so thick as shown here. So now, we want to model this problem. How we do it? Of course, there are many modeling works, but I will present you very simple model shown here. In fact, there are only two variables. The first variable is the concentration of cells called M here, the concentration of monocytes. Monocytes is cells of innate immune response. So we have this M here, and A, this is a concentration of inflammatory cytokines. These are molecules which provoke inflammation. So what is shown here is that this DMDT, so it depends on this cytokines. So this inflammatory molecules, they attract cells. It's the first equation. And the second equation, the cells M, they produce even more these inflammatory molecules. So this is how it works. This is this positive feedback loop between cells and inflammatory molecules. Here it is written just this very simple ordinary differential system of equations, two equations. We can, of course, easily study it. And what we find that there are these different cases we just discussed before, that with respect to the number and the stability of stationary points. In this case, shown here in the left figure, there is only one stationary point, and it is globally asymptotically stable, and the disease does not develop in this case. In this intermediate case shown here, there are two stable stationary points. As before, the first stationary point is disease-free, no disease, and another stable stationary point, responds to the disease development. So this is a bistable case. And to go from disease-free to disease, the perturbation should be sufficiently large because these two stable points are separated by this unstable point. If the perturbation is small, we will return to this first stable point. If the perturbation is large, then we'll go to the second station. And the last case, it is the monostable case. There are two stationary points. This stationary point without disease, it becomes unstable. This other stationary point becomes stable. And even for a small perturbation, we'll go necessarily to the second stationary point. And these three different cases, they depend on the cholesterol level. So we have a good diet, we have low cholesterol level in our blood, we have this left case, no disease. In the second case, if the level of cholesterol is intermediate, we have this bistable case. And in this case, it is possible that nothing happens if the perturbation is small, or it is also possible that atherosclerosis begins to develop if there is a perturbation is sufficiently large. And in this last case, in the right figure, when the level of cholesterol is sufficiently high, the disease will necessarily develop. So this is a very simple model of two ordinary differential equations, but then we introduce these diffusion terms. We obtain this reaction diffusion system of equations, and it is exactly this monotone system of equations for which we can prove the wave existence, the wave stability in the bistable case and in the monostable case, and all these previous results we just discussed in the theoretical part are applicable here. And the conclusion of this study of this atherosclerosis with this reaction diffusion model, that in fact its development is determined by the concentration of low density lipoproteins. If this concentration is low, then we have just a single globally stable point. There's no disease. In the intermediate case, we have two stable points and this bistable wave. And if the concentration is high, then we have the monostable wave. Sorry, and disease will necessarily develop. Sorry. Yes, and there are many generalizations of this model and other modeling approaches. And sorry, since we approach our break, I'll just show you this another modeling, the cell matter modeling. So you see here this blood vessel. Here with this blood vessel walls. And you see this green spots appearing here. These are atherosclerotic plaques, which develop in time, more and more of them, and they change the vessel form, and they and this lumen becomes monero. Sorry, Benjamin. I think it's a good moment to make a break because I need some some water.
Benjamin, you're muted.
Sorry about that. So we're going to take a break to let Vitaly have some water and maybe some coffee, and and so we come back within 10 minutes. So at 11. So be here at 11, and just a little bit after 11, we start again. So
Okay.
Thank you.
11. It's 5 in France.
Yeah, sorry about that. All right. So it's coffee time, water time, whatever you want.
Okay.
Vas-y, je t'ai envoyé un message.
Ah oui, dans le chat. Je sais pas, je vérifie.
Moi aussi, je peux vérifier. Mais...
De toute façon, hein, quand tu arrives à cette heure-ci, c'est trop tard, quoi.
Yes.
Tu veux de l'eau?
Je monte dans la salle. Non, ma puce. Écoute, je trouve pas, mais c'est peut-être un doctorant et doctorante des collègues là qui ont partagé, mais là je connais pas, je vois pas. De toute façon, à cette heure-ci, hein, il va falloir qu'on.
Ouais, on va discuter.
Oui, on discute après, parce que là. D'accord. Ouais, mais il faut expliquer aux gens que on peut pas ouvrir quand on n'est pas sûr et qu'il n'y a pas le prénom nom. Parce qu'il y a eu des perturbateurs qui ont un peu planté les exposés, c'est pour ça. Hein.
Oui, bien sûr.
Le souci, il est là, c'est tout.
Hopefully it will be better now.
Okay. So in the model that you described, the DMDT and has a minus lambda m term, right? For both of them, that seems to suggest that the process is somehow self-limiting.
Well, yes. So
what
this
does that correspond to in physiologically?
Yes, right. Physiologically, it means that cells can die, and this is minus term, and these molecules, they can also be consumed or destroyed or somehow eliminated. This is why there is minus term in the second equation.
Actually, in both equations, right? So both the
in both
information
equations, right? Yes.
Yeah.
So yes. Well, these are these equations with these minus terms here. This is usual situation in biomedical problems because there are some elimination terms usually.
And it has nothing to do with the channel becoming more and more narrow.
No. It's no. In these equations, in these two equations, this question is not taken into account. But well, there are more advanced, more sophisticated models, and in fact, what I showed you just here, another type of modeling where this geometry also is taken into account. We see how it changes in time. Like for example, here you see the channel becomes more narrow. But also my last slide here was also related to that. In fact, it is what is called plug flow interaction. So the channel, the lumen, it is called lumen, this becomes more narrow because of this plug. You see this plug formation here, like shown here. The path with flow becomes more narrow, and there is interaction between flow and the plug. And this, in fact, there is a pressure from the flow on the plug, and the plug can rupture. So this is one of these bad possible consequences of atherosclerosis, that the plug can rupture. And in fact, this is what I was going to tell here, that in fact, either blood coagulation starts here where the plug ruptures, or some pieces of this plug can be removed by flow, go downstream, and can block some vessels downstream. This is also very dangerous. Okay, so shall we begin? Or
Yes, sure. We should start.
Okay.
Please mute yourself. during the talk to let Vitaly has full power to provide his ideas.
Okay, thank you. Then I will continue. And in fact, what we discussed here, that to finish with this part about atherosclerosis, that this atherosclerotic plaque develops. There's blood flow here, and there's some pressure from the blood flow on the plaque. The plaque can rupture. By the way, here, what is shown here in particular, there's what is called fluid structure interaction between fluid and plaque. This become already quite complex models. So the plaque can rupture. It is shown schematically here, and here where it ruptures, it is possible that blood coagulation can start here, and the clot which forms here in this part, it can completely block the vessel, and of course this is very dangerous. So the next part of this application part will be devoted to this blood coagulation and clot growth. So what happens? It's of course it is a very important physiological mechanism, and if there is an injury, then this injury initiates blood coagulation. Blood coagulation, these are chemical reactions, and also blood cells like thrombocytes participate here. And what they do, they just close that injured part, and this allows them to stop bleeding. So this is the role of blood coagulation and clot growth. But there are many pathologies related to this process of blood coagulation and clot growth. In particular, we briefly discussed atherosclerosis, and this is an atherosclerotic plaque. So either clot can come here from outside and block completely the vessel, or it can be formed here and also block the vessel, or it can go elsewhere, and this can lead to heart attack or to stroke, for example. And of course, there are many other different pathologies, in particular deep vein thrombosis and many others. So how it works? In fact, blood coagulation has two main components. These are complex biochemical reactions in blood plasma, which lead to formation of fibrin polymer and also platelet aggregation. And they work together, and this is how the clot is formed. There are many other factors, in particular, blood vessel wall participate in this process with many different regulatory mechanisms, and also blood flow influences clot growth. And again, it's a very big domain with many works and many different modeling approaches. And usually they start with ODE models to describe concentrations of different molecules, in particular thrombin. It's a molecule called also blood factor or enzyme, which plays an important role there in this process. And finally, so here, what we see here, it is a simplified this coagulation cascade, chemical reactions leading to formation of thrombin, and then finally to fibrin polymer. And in fact, this fibrin polymer forms this fibrin clot. And then when we want to model these processes, we can start with ordinary differential equations. Then we consider partial differential equations, in particular these reaction diffusion equations shown here. Sorry for that. Hopefully I will be able to finish this lecture. And as before, we can study these reaction diffusion systems of equations In this particular case, it is a monotone system of equations. It describes propagation of waves, as we discussed before. Then we can, in this, for such models, we can prove the existence and the stability of waves. And in particular, if we measure the concentration of thrombin, it is shown here, in different moments of time. You see here this at the left bottom figure, this thrombin concentration propagates like that. And this is exactly the reaction diffusion wave. And in the vessel, in fact, it is shown here in the bottom right figure. So this rectangular domain is this blood vessel. So the injury is here at the bottom part of the vessel. So here these chemical reactions are initiated and they propagate from the bottom upwards, and they form like a circle shown here. And if we take a cross-section, a vertical cross-section here, we'll obtain exactly this concentration distributions as shown here in the left figure. So what we discussed in this first theoretical part is can be applied here to study this propagation of reaction diffusion waves for this coagulation cascade. And then if we continue even more this simplification of this model, we can reduce this system of equations to just one single equation for the thrombin concentration. And then it's easier to see that in this case, in fact, as before, we can consider monostable case and the bistable case. It appears that for this particular model, not for the model, but the process of blood coagulation, it corresponds to the bistable case. There is this unique wave propagating with some speed c, which can be determined in the experiments and also in modeling. And it appears that the speed of wave propagation of blood coagulation is very important. Here you see some experimental results obtained for hemophilia patients. So hemophilia, it's a disease where blood coagulation is not sufficient. So here you see these curves. These are, well, just to make it simpler, this wave propagation, and you see this in these cases A and B, this wave in fact does not propagate, or it propagates slowly. So if we apply some medicine, then we can restore normal propagation shown here in the figures C and D. So in fact, what we see here is that the speed of wave propagation plays an important role for these blood coagulation problems. Well, I skip that. And as I told you before, each time I use this well occasion to show just a little bit more general models and results. And in particular, what is interesting here is to see how this blood coagulation is influenced by blood flow and by the presence of blood cells, in particular platelets. So for blood flow, if you want to study the influence of blood flow, you can consider in particular Navier-Stokes equations with some additional terms related to this clot. And the main difference here is that you see before we had this, like the concentration was just a circle here because flow comes here from the left, so it changes the form of the clot. And not only it changes the form of the clot, but it can also change the size of the clot. And this is also very important because when we have this clot growth, one of the important questions is this excessive clot formation. So the clot becomes too large, and in fact, it can completely block the vessel. And this depends on many different factors, on chemical reactions, but also on blood flow. So there are some models which allow us to study the clot size and shows this thrombosis, all the things, but I think that I will not discuss in detail now. There are some analytical results, there are some numerical simulations. Like here, I will just show another type of monotone. Spark is treatment. So what I'm showing here is this right figure, the small circles as liquids. So liquids aggregate here at the bottom of the image injury site. At the same time, you see that this blue area here, this chemical declaration X, which lead to production of this type of polymer. You can see this that flow can eliminate some parts of this aggregate shown here. It responds to the experimental observation phase. But indeed it has like that. So here there are some experimental results updated in the liver.
Hi, Vitaly. Excuse me, Vitaly. We have some problem of flow. I don't know.
So it has worked. Shall I continue or
maybe?
So I think because your voice is breaking, I think it's the system cannot sustain the videos and the voice.
Okay,
yes.
then
Yeah,
okay,
so it was very.
I stopped.
Now it's perfect. Now we can hear you well. Yeah.
Okay, so no videos anymore. This is the idea. Okay, let us continue without videos. Okay, so yes, sorry. Okay, I wanted to show you here another video. I will not do it. It's another video. I will do it without video here. In fact, it shows this clot formation in heart. It's a very dangerous process. It's shown here. So in fact, I have a video, but without video, it is called left atrium appendage, shown here. And because of heart fibrillation, so there's a clot forming here in this part. And why it is so dangerous? Because it goes directly from the heart to the brain and provokes stroke. And it's of course, a very important topic, and there are now many modeling results devoted to this question. In particular, some examples are shown here. It's a very complex question because it's all that happens in heart. And there's a very complex hydrodynamics and variable geometry, and on the top of that, this chemical reactions of blood coagulation. This is a very complex but very important problem. Well, then I will not show that. And to finish with this blood coagulation problem, just another application is related to various inflammatory diseases, in particular what is shown here. It is called rheumatoid arthritis. You may know that it's a chronic inflammation, and there are some inflammatory molecules produced this in the inflamed part here, and these molecules also increase blood coagulation, and because of this increased blood coagulation, the risk of cardiovascular diseases strongly increases also. And some modeling are shown here. So I think that this is all for this second part related to blood coagulation. And then the last part, just a minute, I will show you another file. Okay, so the last part is, well, in fact, there are two questions. The first question is about viral infection and immune response, and then the second part will be about neural field models. So again, another application of these reaction diffusion models.
Vitaly, you have to again put your
yes, I will.
Thank you.
Yes.
Thank you. Thanks so much.
Okay. So as before, I will first briefly recall what happens here in what is viral infection and immune response, how it works. So viral particles, they cannot, a virus infected cannot multiply by itself. It penetrates host cells as shown here. And virus, well, it can be DNA or it can be RNA. So are here already inside the host cells. They penetrate the nucleus, enter the host DNA. They begin to multiply, to replicate. And after some time, these new viral particles appear here inside the cell, but then they leave the cell into the extracellular space. And like that, one cell can produce many new particles, many new viruses, hundreds and even thousands. And at the same time, our organism reacts on this viral infection. There are different mechanisms, innate immune response and adaptive immune response. So there are the cells of innate immune response, like macrophages or dendritic cells. They recognize these viruses called antigens, and then they activate then T lymphocytes and B lymphocytes, and T lymphocytes eliminate infected cells, and B lymphocytes produce antibodies, and these antibodies neutralize these viral particles. So this, of course, this is a very short and very simplified presentation of viral infection and immune response. And to study these questions, so it is convenient to consider a simpler case. It's viral infection in cell culture in the beginning without immune response. We have just virus and we have host cells, and we observe how this virus multiplicates in cell culture. And in fact, it is a standard, it is called virus multiplication assay, so multiplicity of infection. And in fact, it is a very common experiment, which is often or practically always used to study viral infection. So if you want to describe this, how virus develops in cell culture, so we can consider this rather simple model of three equations. So the first equation for U are uninfected cells, V is virus, so virus infects this uninfected cells, so this is why we have this minus UV. So uninfected cells U become infected cells called I here, with the same production terms as now UV, it's the same term here, uninfected cells become infected. So then after some time, infected cells can die. This is why we have here minus some constant I. And finally, we have this virus V here. So the diffusion term is because virus can diffuse in the extracellular space. So this second term, it's virus production by infected cells. And this here, this tau, it corresponds to time delay. So in fact, we have here a delay equation for virus production, and virus can also die. So we have this minus V term. So like that, we obtain a delay reaction diffusion system of equations. So, and again, we'll see how we can study it using the methods, the results, or maybe some generalizations of the methods and results we discussed before. So the first simplification, what we can do here, if we assume that beta here is 0, so there is no death of infected cells, then we have the same minus UV here plus UV here. We can take a sum of these two equations, and then the right-hand side disappears, and then we can express we have just the sum of these two concentrations, uninfected cells and infected cells. The sum is constant, so we can in fact eliminate this equation for the unaffected cells. And after that, we'll have only two equations for infected cells and for viruses. Again, we have this delay term, but what is interesting here, that as before, we obtain this monotone system of equations, these two equations here, and therefore we can use also this mathematical approaches we discussed before. We can study existence and stability of waves, though this delay term makes it a little bit more complex. So we have now these two equations, just as before, for infected cells, for viruses, no diffusion for infected cells, diffusion for viruses. And first what we do, we just do some numerical simulations just to see what happens here. And we see exactly this reaction diffusion waves propagating here. This red curves, it's for viruses and green curve for infected cells. So what we can expect here is the existence of solutions in terms of reaction diffusion waves. And this is why now we can study it already mathematically. So we look for solutions in this form as these functions depending on this combination of variables, x minus ct for infected cells, for viruses, and substituting here, we obtain this system of two equations. First order equation for infected cells for the variable w, second order equation for viruses for the function v. What is interesting here is this term, because of time delay, we have here this shifted argument. Instead of, in fact, the space variable now is x minus ct, this variable is called xi. So here, instead of xi, we have xi plus c tau, where tau is time delay, and some values at infinity. So we can now study the existence of solutions like that, and we can study the speed of propagation. Okay, I also, as before, use this occasion to show another method, since it's tutorial. It's a very well-known method to determine the wave speed. In fact, in the monostable case, it is the minimal wave speed. So I present here very shortly this method. And I begin with, to explain it, I begin with a single equation, not two equations as we have there, but just a single reaction diffusion equation without time delay, written here, exactly the same equation we considered before in the theoretical part. And then we linearize this equation about the solution W equals 0. So we substitute the W equals 0, we linearize about the solution, and we obtain this second order linear differential equation written here for u. U second derivative plus cu prime plus f prime 0u equals 0. And since we have here constant coefficients, we can look for the solution in terms just of this, in the form of this decreasing exponential. Lambda here is a positive constant, e to the power minus lambda x. We substitute in this equation, and then we obtain just this second order equation, polynomial with respect to lambda, to the exponent. Here it's equation, and we can solve this equation, and we see here this expression in the square root c squared over 4 minus f prime 0. And I recall that we look for a positive solution lambda. Lambda is written here. So we don't want to have a complex solution, we want to have real solutions. So we impose a condition that this expression is non-negative. And then this means that c squared is larger than or greater than or equal to 4 prime 0, and so we obtain here the minimal value of speed for which this condition is satisfied. So we obtain like that the minimal value of speed C0 equals 2 square roots of f prime of 0. But then, of course, it's not proved that waves exist for all such speeds. We obtained just an estimate. In fact, this C0, it gives a lower estimate of the minimal speed, that we don't know whether we obtain really the minimal speed or it just less than the minimal speed. And then it depends on the equation, on this function f. So in some cases, in particular for the KPP equation, it can be proved that indeed this is the minimal speed. For some other equations, this is not the minimal speed, but the lower bound for the minimal speed. So this is the method. And then we can use the same method for our problem with two equations. Of course, I will not repeat all these calculations. They're quite simple. But if we do them, we obtain this expression at the bottom here for the minimal speed. It is given as a minimum of some function. And then indeed, we compare with numerical simulations, and we obtain that indeed this expression gives us the minimal speed. And then not only in numerical simulations, but we can also prove that for this delay reaction diffusion system of equations, the waves exist for all speeds greater than or equal to this minimal speed. Well, it becomes a little bit more complex because of the time delay, and in fact, the theorem is formulated with some restriction on this time delay tau, that it shouldn't be too large.
What should I think of X as in all of the discussion? The physical space, the physical space X on which you have a you have a diffusion.
The physical space, yes, the physical space, it's here, it's cell culture. So what this means, that we take this solution containing virus and we pour it in like this kind of what is called Petri dish, like in cell culture. So, and you see this small white spots here, it's how, so this, in fact, this white spot, or maybe here it's better to see it on the top figure. So in fact, this viral plaques, they grow in time. So this, in fact, well, this small spots, maybe several millimeters in diameter, but in fact, these are cells died because of virus, and this small and this white spots, they grow in time. So what we see here, it's how this X variable, it's so you can think that we have this white spot here, and this X variable, it's the distribution of this concentration along the diameter of this viral plaque of this white spot. Is it clear?
Okay, so it's not the dish, it's inside the viral.
It's inside this viral plaque. Yes,
yeah.
it's
okay. Thank you. Yeah,
it's not the whole dish, but it's just one part of it.
That's what I wanted to know. Thanks.
Yeah. Okay. And then, like here, you can see some experimental pictures for this. Now, of course, we are all interested in this coronavirus. So this is for this coronavirus infection in the culture of epithelial cells. Epithelial cells is exactly the cells in our lungs. So they study how this viral infection propagates coronavirus in these epithelial cells. And then what is very important in these experimental measurements, it's the total virus concentration shown here in the whole cell culture. And then we can compare with modeling. So what we do, in fact, to compare with these experimental results, we just integrate the solution with respect to space, and like that we obtain the total virus concentration as a function of time. And then, so this is exactly what is shown here in this experimental curves. So we compare these experiments and modeling, and we see that these models describe quite well these experimental results. What is interesting here, that we can identify here in the experiments and in modeling 3 stages of virus progression. So we start. So now this axis, horizontal axis, is time. The vertical axis, it's this integral, the total virus concentration. So in the beginning, virus concentration decreases. Why it decreases? Because it needs some time to replicate in the host cells. From the mathematical point of view, it's exactly this delay term. So if we consider this equations, here we have this delay term. So virus needs some time to be produced in the infected cells. It can be different for different viruses. This experimental results, it is 10 hours. So after 10 hours, virus begin to replicate in the cells. So we see this in the experiments, and in modeling, we need this delay term to describe that. And then when virus begin to reproduce in host cells, we see here its concentration grows very rapidly, kind of explosive growth of virus concentration. And after some time, it stabilizes to this more or less constant value or slowly growing value. And this third stage of infection development, it corresponds exactly to this wave propagation. And the speed of wave propagation determines virus concentration. So this is about how it happens in cell culture. Another interesting question from the mathematical point of view, it's equations with a distributed delay, but I will not discuss today. Yes, so the next step of this modeling, so we introduce now immune response. And as before, I begin with a simple model, 2 ordinary differential equations to explain what happens here. And so these 2 equations, the first one for the virus concentration and the second one for the concentration of immune cells. In the first equation, the first term in the right-hand side here, it's a logistic term. It's how virus is reproduced. And the second term, minus VC, how virus is eliminated by immune cells. And in the second equation for immune cells, so again, this logistic term, how immune cells are reproduced. And we should also remember that immune cells is initiated by, immune response is initiated by virus. This is why this function depends on virus concentration, moreover, with time delay. And also immune cells can be killed by virus. So this, again, this function depends on virus concentration. It's the death of immune cells. You take a simple case without time delay, and we can easily study this ordinary differential system of equations, the stationary points, their stability. And though it's a very simple model, it gives, I think, a rather good understanding of what is happening here. So we have here these several stationary points, and here, stationary points here, so virus concentration either equals 0 here, or it is very small. But here, virus concentration is large, and the concentration of immune cells is small. And these points are stable, in fact. So now what happens? And we start from here. Initially, there are no virus, no immune cells. So we introduce a small initial viral load. So we are somewhere here. So this trajectory can go this way. And in this case, virus is eliminated and the disease is finished. But we can go also this way. And in this case, virus persists and we have either chronic disease or even in the case when immune cells are exhausted, it can be really dangerous and the patient can die. So between these two different cases, how it depends, how we determine which scenario will take place. And in fact, it depends on the initial viral load. If the initial viral load is small, we go this way. So initial V is small, we go this way. Initial V is large, we go this way. So it is very important to decrease initial viral load. And for example, applying this result to this coronavirus epidemics, in the beginning of this epidemics, it was largely discussed where we should wear masks or not. So of course, these masks, they do not protect completely against virus, but they can decrease the initial viral load. And this, as we see, it is very important from the point of view of disease progression. Well, I don't know what is this red line, sorry for that. But then if we continue to study this model, these two equations, under some assumptions, we can reduce it, in fact, to the single equation written here for the virus concentration. And in fact, it is a single delay equation. So what we have here, so this U minus U, this is virus reproduction, and minus FU tau, it's how this virus is eliminated by immune cells. This is a very simple but quite nice model, and I just wanted to show you that even for such simple model, behavior of solutions can be very complex. And in fact, here there is a sequence of period doubling bifurcations, transition to chaos, then order beyond chaos, Sharkovsky sequences, and all this complex behavior can be already obtained in such a simple model. But then, so we have the same term here for virus. So virus reproduction, virus elimination by immune response. But we add now this diffusion term, and like that we obtain this delay reaction diffusion equation. So we can now see apply this our previous results to study this equation. And here, well, I briefly recall, but I will not do it, in fact, about these results about wave existence. What is new here, and this is why I wanted to show this model, that in fact, this function f here can become more complex. Before, we had just a single bistable function like that, but here it can become more complex with several zeros, like shown here, or like shown here. In fact, our function f, it is exactly as shown here. You can see in this figure. So we have here v1 minus v minus f. So this 1 minus v minus f. So the difference is positive here, negative here, positive here. So we have this function like that. Positive here, negative here, positive here. And we can call it monostable, bistable case. Monostable here and bistable here. And in this case, the situation becomes more complex. And in fact, depending on parameters, we can have just a single wave propagating like shown here in the bottom left figure, or we can have two consecutive waves propagating with different speeds as it is shown in the right figure. And why it is interesting for our applications? Because what is happening here, and it describes how this infection spreads in the tissues or organs of our organism. So there's these three different cases shown here. In the first case, this green curve, So this wave of viral infection propagates with some speed, and behind the wave, we have this low level of infection. Another possibility is that there is this red curve. So again, there is infection wave propagates, and we have high infection concentration behind the wave. And finally, it is also possible that we have two waves propagating with different speeds. First, we have this fast wave, such that there is low infection behind the wave, but then there is another wave, such that behind the second wave, there is this high infection concentration. So this example describes these different regimes of infection spreading in the tissues of the organism. Well, then I have several examples, numerical simulations showing different pulses and waves and emergence of what is called virus quasi-species and evolution of virus quasi-species, but I will not stop on that now. And different waves, periodic waves, and quite complex and quite interesting behavior, because equations here can become, well, with time delay or non-local equations. So there are many generalizations and developments of these equations and of these results. And last several minutes, I will spend for the last problem I would like to discuss. It's about brain modeling, also some applications and some developments of these mathematical results. So as before, I will begin with a short biological introduction, and just to mention that it is a very active and very interesting, very complex field of research. And now in neuroscience, what is very important, it is the notion of connectome. And connectome means that brain functions, it's not in fact localized functions, by distributed functions, networks, and shown here for each of our functions like speech or other activities. It's not just a single brain area, but the whole network which is concerned. It's also shown here for the example of language. For example, in the experiments or in our everyday life, when we have this image and we want to pronounce the word, in this case it is cat. So it is known that, in fact, it is a very complex process which contains, now they separate 6 stages of this pronunciation. And for each stage, there are special brain networks which are concerned, and they are shown here in these consecutive figures. What is also interesting, well, I don't know whether I can take a risk to show you this video, but in fact, what is also observed here, it's wave propagation in the cortex, shown here. These are experimental results. And in fact, there are different waves with different frequencies. And here it's another example. It's not plane waves, but rotating waves shown here. And here is another data from EEG waves. Again, these plane waves can propagate like that, like shown here, or it can be rotating waves like shown here. And then, of course, if we discuss modeling, there are different modeling approaches, and one of them, it is what is called neural field models. In fact, these are integral differential equations, and there are many different models. But what I want to show here is, and also to use this occasion, is another, mathematical method or mathematical approach to study these problems. And this is related just to stability of solutions, not to stability of waves or pulses, as we discussed before, but to stability of homogeneous in space stationary solutions. And this is a quite simple mathematical method. So if we look at this equation, we can verify that u equal 1 is a stationary solution of this equation, and we assume that the integral of phi equal 1. So we substitute u equal 1 here. So the integral of phi equal 1. So this is 0. So everything disappears. So u equal 1 is a stationary solution of this equation. We can linearize our equation about the stationary solution, and then we obtain this eigenvalue problem. And then we apply the Fourier transform. We obtain explicitly the eigenvalues for this problem. And like that, we can study the stability of this solution u equal 1. And it appears that if we consider this equation, then for the local equation, when we have here 1 minus u, the solution u equal 1 is always stable. But if we have this non-local equation with this integral term, then the solution can lose its stability, and there are these periodic structures can be like that here. And in dynamics, we see that this wave propagation now, but now behind the wave, we have this periodic structures appearing here.
Sorry, Vital, just very,
yes.
You said it can. What is the parameter? So
yes, if we consider this equation, so we can easily obtain the stability conditions here. In fact, for this equation, of course, it depends on the equations, but for this example, this condition is given here. It depends on the diffusion coefficient, which is here. It depends on the n. Well, it is not explained here. N, in fact, is the support of this kernel phi. So there are several parameters, but in this particular case, it can be.
Okay, thank you.
This is clear. Oh. Yes, but then just another equation, a little bit more complex, with two integral terms, one with sign plus, another with sign minus. Why it is interesting? Because there are different structures which can appear here, in particular what is called periodic waves shown here, and other different waves. So the behavior can become quite complex and quite interesting. And my last example is shown here. It's exactly about this periodic waves in this one of these neural field models. You see that it's like a sinus which moves like that here. So I think that I can stop on that. And well, the conclusions which I would like to present you here is that physiological systems are often characterized by the presence of stable equilibria. And the stable equilibria can correspond, for example, to disease-free, like, for example, we discussed in atherosclerosis, like the unique globally stable stationary solution, no disease develops, or it can be like in this neural field models, it can be a quiescent state. So this stable equilibria, but if there's some activation of these physiological processes, and by activation we can understand different things. It can be either disease development, for example, or it can be like an excitable media, brain activity, and other things. So then this stable equilibria can lose its stability. Some other solutions can appear, either equilibria or other structures, spatial or temporal structures can appear. And if there are several of these equilibria, then the transition between these equilibria are described by reaction diffusion waves. So I think I will stop on there.